You can restart the thing there. So let's go back to talking a bit about the retina here. This, here's a cone and here's a rod. This one's called a cone because it looked like a cone under a microscope to somebody, and they called this a rod because it didn't quite look like the cone. It was more straight up, so they called that a rod. The rods are more effective under low light conditions. People under low light tend to be able to detect bluer lights better. That's the reason that blue lights were originally forbidden for headlights, and they use red lights when you're going to be in a situation at night where you want to preserve your dark adaptations. So bomber pilots in World War II, they would use red light, turn on a red light to do something, turn it back off so they could see well at night. Nowadays people have sort of forgotten their psychology and perception, and they're allowing blue lights, blue headlights on some vehicles that hurt people's dark adaptation. If the pigment, so here's the pigments in there. If the pigment is blue, what color is this cone detecting, you think? If the pigment looks blue. What's it absorbing?
Short wavelengths.
Right, so it's absorbing, oh, it's reflecting the short wavelengths. What's it absorbing? It's absorbing the long wavelengths. So this cone is detecting red. All right, so this is a red detecting cone. Remember, the color that you see is what something is not. If you were trying to grow grass indoors, don't know why you'd want to do that, but if you're trying to grow grass indoors, what color light would be the least effective? Green, right? Because we know grass reflects the green light. It's not absorbing the green light. So other colors, red would help with let your plants grow. It's reflecting green because you see the green reflecting off the grass. That's the one color it's not using. All right? So this cell here would look blue if it's detecting red. I don't even want that distracting thing, so let me bring this display up. All right, so this is the picture from Gelb, which is a little bit simplified. We'll go over it in a bit. And this one, this cell is more likely detecting a red. All right, I mean detecting green because it's reflecting red. Now these cells are detecting it. Remember, the light is being absorbed, converting the pigment into the bleached form, and energy has to be used to recreate the pigment so that you can see again. So each time that light binds chemically with it, it's going to cause spike events to happen. It's going to send spikes from the ganglion cells down their axons. These are the axons that are going to the optic nerve and leaving the eye through what's the blind spot. There are also these horizontal cells and amacrine cells that are running this way. These are the bipolar cells. They've got you know, cytoplasm and mitochondria and cell and the chromosomes in there in the cell nucleus, and all those things are in there, just like the other nerve cells. But these are the specialized ones that contain the pigments that are detecting the light. And when you detect the light, they're becoming bleached, and we have to unbleach them. So now let's look at the opponent process theory of color perception. Let me turn off the, and we'll see what's the, actually I'm going to go ahead and do that one too. This one. All right, so here's the diagram from Gelb, and the diagram, they're intending this red that they've drawn around the outside of it to tell us that this cone is detecting red light, so they put red around it to indicate that it's the red detector. So notice they said the long wavelength cone, it responds well to red or yellow, all right? So this cone, you can see here they put green inside. Looks like Cal told them, I want you to put kind of green little bodies there that are absorbing pigments, absorbing light. Here's our bipolar cell. We've got an excitatory synapse on this cell. This bipolar cell here is excited by red and inhibited by green, and they put green around the outside here to indicate that the cell here is responding to green. The pigment inside here should look red because it's absorbing the green light. So these pigments ought to look red that are in there. But the color around the outside is what they're saying. So this one is a medium wavelength cone. It's responding best to green light and less to the red. And so this one is inhibiting this cell. This one's exciting this cell. Everybody clear on that? So this cell is a red detector. It allows you to perceive red. Yes?
So in that diagram, where are the eyes exactly?
Where is the eye? This is in the back of the eye. This is the retina. So here's our retina right here, right? This is the back of the eyeball. The light comes in through the pupil and it goes through the retina. So in this diagram, it's coming from this direction, past the blood vessels, past these axons, through these ganglion cells and the bipolar cells, and then it strikes here. All right, and it's absorbed. All right, and so that's this. I've used the same alignment here as here. So the light is coming from this direction, and it's in the. You got the geometry?
Yeah.
So that's in the back of the cell, and the light is coming in the picture from below, lighting it up. And these things are going to attach to the ganglion cells, which are going to send their axons that go out through the optic nerve, and then to the optic chiasm, and then up to the eventually to the occipital cortex. All right, everything clear there? So this bipolar cell right here, they've written on here to the ganglion cell. And so here's I've drawn in the ganglion cell, and then they're sending their axons up to the optic nerve, all right? So this one is responding to red. Now what we're going to do in the demonstration is we're going to try to, so if I light both of these up, are you going to see red or green? If I shine both red and green light together, which white light is composed of red and green, right? Red, orange, yellow, green, blue, indigo, and violet. Where's the indigo, incidentally? You guys ever wonder about that? Well, Newton wanted to have 7 colors. So when he was separating the colors out, he said, you know, there's got to be 7. There's 7 notes of the musical scale. There's 7 days in the week. There's 7 deadly sins. Snow White has 7 dwarfs. You know, you got a lot of 7s. So he wanted to have 7 colors. So that's where the indigo comes from, all right. But if I shine red and green together, people don't see both red and green. They see white. Okay? And so this one's excitatory and this one's inhibitory, so if I shine both red and green, the person doesn't see either red or green. They cancel each other out. So we've got an excitatory synapse and an inhibitory synapse, so they cancel each other out, and you're going to see white if I put both red. On the other hand, if I have red light and no green light, then the person is going to see red optimally. Okay? And over here we've got the same kind of thing going on for blue and yellow, and we can have blue versus red, because there's a lot of things you could have with inhibitory synapses, but let's focus on this one for our demonstration, all right? Now, if we keep looking at green, if I keep showing you green light, I'm going to poop out these detectors. What do I mean by poop them out? We're going to bleach the pigments faster than the body can regenerate. So if you run down the road, you get pooped out. Why? Because you're breathing really hard, you're using up all the fuel that's in your muscle cells. Your bloodstream has to bring more fuel. You're generating lactic acid. You got too much carbon dioxide in the blood. You get tired, right? You have to stop and rest. And the body can regenerate and send some more sugar, right, so that you get your energy back again. So vision has to work the same way. For you to see, you have to be able to regenerate it. So we're going to poop out the green and then shine white light, which is both red and green. So if I show you a lot of green light, you're going to perceive red because of the release from the inhibition. Everybody clear on that? So that's what we're going to do. Everybody clear on the theory of what's going to happen? So we're going to do an afterimage. I want you to look at the dot with, I wish this thing was brighter. All right, we'll see what we can do here. This doesn't work. I've got one posted at the URL. Look at the dot, the white dot in the middle of that. And just keep looking at that. Don't look around at other things. Just keep staring at that dot. And as I say, I wish it was brighter. There's one for you to look at in the book. In some cases, it might be a cartoon, or it might be a Mona Lisa picture, it might be Elvis Presley, who knows, or maybe Marilyn Monroe in different editions of the book. This case is the flag. Keep looking at the dot. Now what's going on is we're pooping out the pigments that would look red, but that are detecting green. So the green detecting pigments are being bleached. Keep looking at the dot. Don't wander, don't let your eyes wander around, just keep looking at the dot. And it should start to kind of shimmer a little bit, the image. Keep looking at the dot. Just keep staring at the dot. Don't let your eyes wander, but there's also like a red scene over there. We're pooping out the detection of red, which is an opponent with blue. But keep looking at the dot. Just keep looking at the dot. Look at the dot. Stare at the dot. We're bleaching the pigments that detect green. Those pigments are becoming bleached, and they're causing spike events to happen that allow you to see the green. If we could poop them out completely, you'd stop seeing green. When something is stabilized on the retina, now you can't perfectly stabilize your retina unless we can put curare in your eyes, because your eyes are moving a little bit. But I want you to not let it happen voluntarily. Right? So your eyes are moving around a little bit, but if you stabilize something on the retina, it will disappear. That's why those blood vessels disappear. They're stabilized on the retina, and eventually they just go out. So keep looking at the dot. It's starting to shimmer. So the idea is we're trying to bleach the retina faster than the body can regenerate it by continuously presenting that same green color and trying to keep it on the same spot. Now it's a spot on the retina that's getting bleached. Not all of your retina is being bleached, just part of the retina, the part that sees this flag. All right, well, let's see if that works. All right, now look at the white sheet here. What do you see? So hopefully you see red stripes now where the green had been, and you see white where the black had been, and you might see a blue field where the red had been. All right, so that's an example of an afterimage. We did it in both eyes. For your homework assignment, you're going to get an afterimage in one eye. All right. All right, if that didn't work for you, how many did it work for you? You got able to see that? Looks like most people. If you didn't see that, look at the one in the book. Shine a bright light on it in the book and follow the instructions there in Gelb. Now things aren't quite as simple. I started with the analogies that you increase the brightness, increase the intensity of light, and it looks brighter, right? But that's not the whole story. It's more complicated by a couple levels. And so we're going to learn a little bit more. Normally, objects don't change color, right? So if you think about it, that's a funny thing. You're used to thinking of it this way, that the object doesn't change color because color is a property of an object. Now, I should have worn a red sweater or something with some color in it. But I have some shoes. I have some black pants here. All right. The pants are black. That means they're reflecting very little light. If I take them outdoors, the amount of light that comes from the pants to my eye is more than 10,000 times greater. So why don't they change color from black to white when I take them outdoors? So if the eye was just like a light meter, when I go outdoors, they should change color, shouldn't they? Because the light that's being detected by the eye is drastically changing. So the question is, why is it that objects stay the same color even though illumination is changing? So if I go outdoors, the pants don't change from black to white. That's a problem to be explained. Now objects do change color, as we know from the Gelb illusion. Now, the Gelb illusion is the one that's kind of hard for me to set up in here, but you can do some things that are sort of like it. Ideally, we would have no light coming from the windows, and in this room, I'd need to bring in blankets and cover them up. It'd be too hard. We need a curtain across the front. We need a projector where the camera is, and we would put a square hanging on a thread between the two curtains. So let me diagram how it's going to look from here's how you do the Gelb illusion if you want to do a really good job of it. Here's the audience in their seats. You have a curtain across here, and you have a projector. And the projector is lighting up a square in a darkened room. So we turn off all the lights in the room, we light up a projector, and it lights up that square, and the audience is sitting, out here. And they see the square in the dark room. And they look at it, and it looks like it's white. So the square in the dark room, in the Gelb illusion, the audience says, you say, what color is it? They say it's white. I say, okay, now I'm going to turn on the room lights. We turn on the lights. That increases the illumination, increases everything, right? And now the audience says it's black. So it changes from white to black. The same object changes color. And that's also puzzling because we have just increased the illumination and made it darker. So it got darker when we increased the light. So that is a puzzle. It's called the Gelb illusion. And it tells you that sensations of color are not just judgments of luminance. So I'm going to need to tell you a bit about luminance and illumination and reflectance. So I have to talk a bit about the physics of the situation a little more on the physics here. All right, here's our source of light. And I can adjust it, and it's the illumination. I is equal to illumination. That's the light that we shine on something. So we've got a patch here of color of some kind. I don't remember if I think I might have asked this already, and there might have been an art major, but who knows if they're attending. Is there an art major in here, fine arts? Okay. And you've probably been sent over to the art supply store and looked at things there, right? They have a lot of good stuff for artists. Pens and pencils and paints and paper and all kind of good stuff. And patches of color, standard colors, right? Munsell colors, did they send you for the different Munsell coloring system? Maybe you bought some colors. So here's a patch of color. And these come in different kinds of hues, patches of color. Colors are, again, pigments. So when you're mixing paints, you're mixing pigments. So we're mixing things that absorb light. When we mix colors together in painting, we're adding pigments together. If you add more pigments together, it gets darker, right? Mix colors, it gets darker. When you add lights together, it gets brighter. All the lights together gets white. Remember, we can separate them out and then Newton combined them back together again? But the pigments, when you add pigments together, you're subtracting light because pigments are absorbers of light. So the more, if the pigments were perfect, if you added them all together, you'd get black. If you add all the lights together, perfect, you get white. So some of the light is being absorbed and it's being changed into something else like heat or chemical energy. In the case of our pigments in the eye, it's bleaching, it's making a chemical reaction. In the case of a patch of color, I could take them up on the top of the mountain where there's some snow on the ground. Is there any snow left up on the top of Wallach? I doubt it this time of year, but who knows. There was some there earlier. But imagine there's a place where there's snow on the ground. We could take the colors out and put them on the ground, on the snow, and let the sun shine on them. They will sink into the snow to show that they're absorbing heat and heating and warming up. Which one is going to sink the most? Black. Which one is going to sink the least? Well, if I had a mirrored one, so like a mylar that I cut out, perfect mirror, it's going to reflect almost all the light. But white, for a flat color, white is reflecting all the colors back. So it is going to sink the least of the Munsell colors. And a gray is going to sink somewhere in between. So black is going to sink the most, white is going to sink the least, and the other colors, depending on the amount of energy in them, they're going to sink different amounts. All right? So here's a pigmented surface. It's reflecting some of it, but it's absorbing some, which means it's warming up. And some of it's reflecting off. R is a property of an object, its reflectance. Now, over here we have an eye that's looking at this thing. The only way for you to see something is for light to bounce off it and go into your eye and be detected there. So the light that goes into your eye, that's luminance. That's the light that comes off the object that you actually can see. So we have the light that's on the object, that's illumination. We have the reflectance of the object. That's the property of the object. That's actual physical property of the object. And you have the light that goes to the eye. That's luminance, the light that goes to the eye. Now, luminance is equal to illumination times reflectance. And reflectance is expressed as a proportion of the light and also as a function of the wavelength of the light. So different Munsell chips are going to have different reflectance patterns. A pure color, if we could get a pure paint that reflected all the red light and absorbed everything else, right? It would absorb everything except, let's say, 650 nanometers. 650 nanometers, those of you who played with a red laser light, most of them are about 650 nanometers. So that particular red is, a laser produces a single wavelength of light. So if you do a Fourier analysis, one thing about a laser, if it's perfectly made, it's producing just one wavelength. It's a very simple picture. And if you could get a perfect paint, it would have this reflectance pattern. It would reflect 650 and nothing else. But most surfaces are mixtures of things, like her sweater that's purple there. It's reflecting a lot of different things. And so many objects, so here's our red up here. So somewhere up around 700, and down here's our 350 or so. So here's our red, orange, yellow, green, blue, indigo, and violet. So here's our colors laid out here. Something that reflects mostly up here is something you're going to call red. Something that reflects mostly down here is going to be purple or blue, or something in the middle, green, right? So red, orange, and something that reflects mostly in here is going to be something that's yellow. Now the objects I'm going to be talking about now are called neutral colors. They have reflectance that's equal all the way across the spectrum. These are the so-called neutral colors. And an object like these pants reflect about 80% of the light that's shining on them. I mean 20% of the light that's shining on them. So 0.2 would be something that under normal conditions you'd describe as black. And something that's 0.8, that's 80%, would be like a sheet of paper. So the blackboard here is up close to 80%. Sheet of paper like this is reflecting maybe 80% of the light that's shining on it. A white sheet of paper, this doesn't look like it's off white, but maybe I've got the white sheet of paper there for you. White paper, about 80% black chip. And it's reflecting all across the spectrum equal amounts. These are the so-called neutral colors. So white, under normal conditions, and black, under normal conditions. But it still isn't as simple as that. So, one of the things you realize, let me review what I just said and I'll come back to Wallach here in a second. The Gelb illusion makes you realize that color is not really a property of an object, it's a sensation. Reflectance is a property of an object. Luminance is the light from an object that goes to the detector or to the eye. This is a physical quantity. Illumination is the light that's on the object, that we're shining onto the object. Reflectance is the proportion of light of each wavelength that reflects back off. And the neutral colors, so the various hues, and so objects differ not just pure hues, but they differ in other qualities. In music, you'd call it timbre, to talk about other qualities of tone produced by complex overtones that are in the waveform, besides pitch. And in color, you talk about saturation and other kinds of qualities. We've gotten pretty good at computer software to simulate, to create the little, well, anyway, I'll get into that a little bit more. So reflectance, that when it's equal at all wavelengths, so the amount of light at all wavelengths, we're talking about the neutral colors. So if the light is equal, we're talking about the scale of whites to grays to black. Are you familiar with HTML? How many are familiar with HTML, the coding of color in HTML to display on a computer screen? Nobody knows HTML. Well, colors are represented on, okay, so the colors are represented in red, green, and blue in HTML. And they use a hexadecimal code, which means that each number can vary from 0, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. So you have 16 possible levels from 1 up to 15. And then you have 2 digits possible for each of these. So they can go from 0, 0 up to FF, which is 255. And so the color, if the red, green, and blue are all equal, you have 0, 0, 0, 0, 0, 0, that's white. All right? And then FF 0, 0, 0, 0, that's red. And FF, 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 that's, that's white. 0, 0 is black. That means no light, right? So all zeros is black. And all F's, that's 255, 255, and 255. And if you vary keeping the numbers the same, so C, 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 or 6, 6, 6, 6, 6, 6, those are the 255 shades of gray. Nobody's seen the movie or read the book, right? On HTML, you get more than you get 255 shades of gray instead of the 50. Right? So HTML is better than movies out of the car. You get you more. Okay, that was a long way to go for that joke. It's a good joke if you knew HTML, though. You liked it, right? You know, there's a little HTML. All right, so he liked it. Okay. When you take your HTML, you'll like that joke better. But that is how you do it. So go ahead and write in your HTML, you know, background color or font color or whatever it is equal to FF0000 or 00FF00. That would be green, or 0000FF. That would be your blue. And you can reconstruct all the things you can construct on a computer screen are constructed from these three colors, because those are the only three colors that are there. So everything you see, all the other colors are created by combinations of 255 levels of each of three. There's only three different kinds of color elements on a computer screen. All right, let's take a look at Wallach's theory now to explain the Gelb illusion and color constancy. Okay. His theory says this. Here's his theory of neutral colors. He also has a theory of hue color perception, and if you like the topic of color perception, or and maybe the fine arts people might be interested in that, take a course in sensation perception. As I mentioned before, Eriko Self is the chair of the Psych department. Her field is color vision. She might be running experiments this semester. Some of you might have been in, where you sign up and go participate in color experiments. So Wallach has a theory of hue perception. We're just going to deal with his theory of neutral colors. And his theory of neutral colors says this, that the color is equal to a function of the luminance from the center to the luminance from the surround. So it's a ratio of luminance of the object to the background, or in his study to the center and the surround. And he does this experiment. He uses episcotistors. That's an old-fashioned way of manipulating intensity of light. Today they do these with display screens like for computers. But in the old days they used to use episcotistors. Those were projectors that had fan blades that would rotate, and you could vary the width of the fan blade. And as they rotate, people will combine them so you could control by the percentage of light being let through. He has four projectors. One projector lights up just the disc over here, and another projector, it lights up only the annulus, or the ring. So it's got a slide that it does not light up the center. So he can vary the luminance of center, we'll call this luminance of center 1, and luminance of surround 1. So this outside part is called the, we'll call it the annulus, or the ring, or the surround, or the background, I guess is the way it's labeled there. And this is the center, or the object that we're talking about, or the disc. I might use those words when talking about the disc. So one projector is lighting up the center, one of them is lighting up the surround, and he's got his episcotistors. I'll write that word out for you here. And the episcotistors are fan blades that spin around and allow him to manipulate the luminance that the projectors are lighting up. You can remember this because it rhymes with "he kissed your sister." Episcotistor. The person I took intro psych from said he would never forget an episcotistor because he was working in Wallach's lab as a grad student, stuck his hand in there, and you know, any blade that's spinning around disappears. You may forget that it's moving. It's in there. He got injured by an episcotistor. That was Alan Verducci. Has a theory of happiness I'll tell you about later. At any rate, the fan blades that are spinning around allow him to manipulate the luminance of the center and the luminance of the surround. And then he's got one over here. He's got another pair. So he's got another projector, and so he manipulates the surround over here on this side. So this projector is lighting up everything except the center. So this is the luminance of surround 2, and the subject's task is to adjust the episcotistor by turning a knob to match the color of the center 2 to the center 1. So the subject adjusts the episcotister over here which basically manipulates LC2, the luminance of center 2, so that the color of the center 1 is equal to the color of center 2. Everybody got the task? So he's able to manipulate, he has large backgrounds and large centers, and he adjusts 3 of them, 3 independent variables. The background on the left, the center on the left, the background on the right, and the subject gets to adjust this projector over here that lights up the center disk so as the colors will match. So he's able to make the colors match. And he finds that if he sets this to 100 and this to 50, and he sets this to 50, what does the subject set the center to over on the right?
25.
25. Because his theory is the color is a function of the ratio. Not the luminance, but the ratio of the luminance of the center to the surround. So the colors match when the ratios are equal. So in this case, the subject would set this to 25. Turn off the 2 backgrounds. The subject says, oh no, they're not equal to each other anymore. All right? Turn back on the backgrounds. Now they match. Set the background to 50 and the center to 50. Then the subject sets the center to 50. All right? So if the ratio is 1 in both cases. Now, most of you are familiar with this. You've got a device at home, most of you, probably all of you, that takes advantage of this. If it weren't for this property of human vision is different from a light meter. If it was a light meter, that's one of the reasons that old-fashioned photographers had to carry around a light meter with them, because the human eye is not a light meter at all. People are deceived by what they see visually. It is not measuring physically what's happening. Nowadays they build the light meter right into your camera, so you don't even realize there's a light meter in there. It's measuring the amount of light, deciding whether to flash, doing all kinds of things for you. Deciding how much time to let the light in, how wide to open the aperture. It's got all sorts of automatic, it's got a computer built in to fix everything for you. But you have a device that takes advantage of this. This device, the only way for you to see black, if you want to see black on here, here's how to see black. Wait a minute, yeah. I want to get a good black here. Let's make a good black. So here you'd be seeing black on this center. But notice this thing is giving off light. If I turn off the background, what is it going to look like? A glowing moon in the night. It's going to look white. If I want it to look black, I have to turn on the background. You have a device at home that the only way for you to see black on this device is to turn it on and have it give off light. What's the device? So for you, it actually has to get brighter for you to see black. It gives off more light when you see black than when it's, right? And you're not doing experiments at home, but maybe you are. What's this device that works on this principle that allows you to see black by actually giving off more light?
Television.
The television. She gave it away to everybody. She couldn't wait. She can't resist giving the answer if she knows it. How many knew it was a television? Hopefully you know. A computer screen would do the same thing because you can see anything. All right, now when you look at the moon at night, the moon looks like a headlight, right? It looks like it's a source of light. What color is moon dirt? It's gray. But it looks like a source of light at night against the dark background. But in the daytime, the moon looks like a dish, right? Different color, not like a source of light. It's a lot less. And if you get some of that same dirt and look at it, it looks like charcoal. So moon dirt, if you've seen some in an exhibit somewhere, looks like charcoal. So the theory, Wallach's theory of color constancy is that the average background, when I go outdoors, the stuff that's in the background hasn't changed. You will have color constancy when the source of illumination of the object is the same as the source of illumination of the background, because then the ratio of light from the object to the background will be the same. And a way to create an illusion, like in the Gelb illusion, is to have a different source of illumination for the object in the background. So now that you know Wallach's theory, you should be able to explain the Gelb illusion. You should be able to explain color constancy and when it happens, when you get illusion, when you get constancy, and you should be able to explain Wallach's experiment. If I set this to 100, what would they set the center to over here? If this color has got to match this one? Would be 10, because 20 divided by 200 is the same as 10 divided by 100. As long as the ratio of the center to the surround is the same, the colors are going to look the same according to Wallach's experiment. It works over a range of about 4 to 1, over the range of things varying from my pants to a sheet of paper. It breaks down for more extreme, and as we'll see, there's another consideration. So it's even more complicated than that. So vision is not just a detector of light, and it's got a ratio built in, and there's another complication that you'll see in the videos. All right, so we'll see you next time.